osmosis in living cells. If it's your first time to come across this channel, please subscribe, like, comment, and share, and don't forget to hit the notification bell. So, what is osmosis? What is osmosis? Osmosis is the movement of water or solvent molecules from a region of high water potential to a region of low water potential through a partially permeable membrane. So we shall look at the meaning of each of the keywords in the definition of osmosis. One, we shall understand the meaning of solvent molecules. The next one, we shall also understand the meaning of high water potential. Then we should also understand the meaning of low water potential. Then the meaning of a partially permeable membrane. So when we talk about solvent molecules, we just can understand the meaning of a solvent. A solvent is a substance that dissolves a solid, and a solid is a substance that dissolves in a solvent. An example of a solvent is water. Example of a solvent. We have water. We also have ethanol as an example of a solvent, but this one here is a solvent for organic compounds. Then we have water, a solvent for ionic compounds. So that is for solvent molecules. Now, what is the meaning of high water potential? High water potential means an area that is having more water molecules. Then, low water potential means an area that is having little water molecules. So, how about a partially permeable membrane? A partially permeable membrane is one. So, let us look at partially permeable membrane. Partially permeable membrane allows movement of some molecules, but does not allow others. Take for example the cell membrane. A cell membrane is an example of a partially permeable membrane. Can allow the movement of solvent molecules, whereas it can not allow movement of some other substances like the proteins through it. In other words, larger molecules. So that is a partially permeable membrane, allows movement of some molecules and does not allow others. So we can call it a partially permeable membrane, or we can also term it as a semi-permeable membrane. So if we have, let me have an illustration. If we have this as our membrane, so in our membrane here, we have areas where we can have the exchange of the materials, and then we have two given regions that are separated by the membrane. So if we have this area A, then we have another area B. We shall be having one area which is having high water potential. Take for example, if we have A having high water potential, and then we have B having low water potential. So we shall have the water molecules move from a region of high water potential to a region of low water potential. For high water potential, it is in a dilute solution. Then where we have low water potential is in a concentrated solution. So in a dilute solution you have more water, then in a concentrated solution you have little water. So the difference now in the water potential is what we shall be calling the water potential gradient. So we have what we call a water potential gradient. So water is able to move from one region to another because of a water potential gradient. One region having high water potential, then another region having low water potential. So here is our membrane, which is partially permeable, can allow some materials, substances, or molecules to pass through it, whereas it does not allow others. So let us look at the concept of osmosis in a living cell. Take for example an animal cell. What happens if you put an animal cell? So we look at osmosis in an animal cell. If you have a beaker and in your beaker you are having pure water, then in the beaker you put an animal cell inside that beaker. What is going to happen to the animal cell? The animal cell inside it is having low water potential, whereas now the outside of it there is high water potential. So what do we expect? We expect movement of the water molecules from outside the animal cell into the animal cell. Then what will happen is that the intake of the water molecule is going to make the animal cell to burst. So after a short while we expect the animal cell to burst because its membrane does not resist the pressure that will be inside the animal cell. So the animal cell bursts in pure water. So if you have pure water, the cell bursts. How about if you have the animal cell in a concentrated solution. In a concentrated solution. So we have another example. You have the animal cell in a concentrated solution. In a concentrated solution, the cell is going to shrink. The cell will shrink because water is being lost from inside the cell through the cell membrane into the outside solution that is having a low water potential. Actually, the one that is having a low water potential. That is from a dilute to a concentrated. So that is for plant cell. In pure water, the cell will burst, and then in a concentrated solution, the cell is going to shrink. Let us summarize with what happens with a plant cell. Osmosis in a plant cell. So if you put a plant cell in a dilute solution, what we also call hypotonic solution. A plant cell in hypotonic solution is going to absorb the water, and after absorbing the water, it is going to become turgid. Turgid means the plant cell is going to become firm. So this absorption of water by the plant cell is important in a way that the plant can gain support. For example, plants with the weak stems. When they absorb the water osmotically, they are able to have the support when the cells become turgid or when the cells become firm. So if you have a plant cell, that is the cell membrane, we have the vacuole, and then we have the nucleus. So we have the cell wall, that's the membrane. We have the cell vacuole, and then we have the nucleus. Don't forget we have not included all the cell organelles in this plant cell. So the cell wall is fully permeable. It's a fully permeable membrane. Then the cell membrane here is partially permeable. So this one here allows some molecules, whereas does not allow other molecules. So in a dilute solution, the water molecules is going to flow, move past the cell wall into the cells through the cell membrane. Then the cytoplasm. So here we have the cytoplasm. So the cytoplasm is going to push, and then the cell membrane will press against the cell wall, and we shall develop what we call a sugar pressure. So because of the sugar pressure, cell contents press against the cell wall, and the cell becomes firm, or the cell becomes turgid. This is when you have a plant cell placed in a dilute solution. The other aspect is when you have a plant cell placed in a concentrated solution. A concentrated solution. what we also call hypertonic solution. So if you have a plant cell in a concentrated solution, the water molecules is going to enter is going to leave the plant cell. From the plant cell, water molecules will leave and go into the surrounding, of which the surrounding is the concentrated solution. The plant cell is going to lose the water molecules. Then the cell membrane is going to pull away from the cell wall. So if the cell membrane pulls away from the cell wall, then the plant cell is going to become plasmolysed. It is going to undergo what we call plasmolysis. Plasmolysis is when the cell membrane pulls away from the cell wall of the plant cell. So that is as a result of loss of water molecules from inside the cell to a concentrated solution. So as we wind up our lesson, we have looked at the definition of osmosis, which have also looked at the different meanings of the terms used in the definition of osmosis. We have looked at osmosis in an animal cell, where we looked at what happens when you put an animal cell in pure water, and then when you put an animal cell in a concentrated solution. Then we have also looked at osmosis in a plant cell. What happens if you put a plant cell in a dilute solution, or what we call hypotonic solution, and then what happens if you put a plant cell in a concentrated solution, or what we call hypertonic solution. Actually, finally, we have what we call isotonic solution. If you put a plant cell in isotonic solution has got the same concentration as the concentration inside the cell. So there will be no change in the shape of the cell. So thank you for watching this video. Please like, subscribe, comment and share the videos, and don't forget to hit the